Bismillahirrahmanirrahim. Dear fellows, Assalamu alaikum. In our previous lecture of gene regulation in bacteria, we have studied the basic regulatory mechanism of gene expression. Now you are familiar with the basic mechanism that gene expression is regulated by some repressor proteins, some activator proteins, some inhibitor proteins, co-repressor molecules and some inducer molecules. This was the basic mechanism of gene expression. Now we will turn our attention to the example of the gene regulation found in E. coli, in Escherichia coli bacteria, and the genes which is responsible for the synthesis of lactose utilizing enzyme. But before examples, we are going to study a basic mechanism of enzyme adaptation. What is enzyme adaptation? Enzyme adaptation refers to the observation that a particular enzyme appears within a cell only when then the cell has been exposed to the substrate for the enzyme. Mean if a substrate is present in their environment, then that cell will produce that enzyme to utilize the substrate for their metabolism. And when a bacterium is not exposed to that substrate for that particular substrate, then it does not make the enzymes needed to metabolize that substrate. To investigate this phenomenon, Jacob and Monod, they studied the lactose metabolism of bacteria. And what was their observation? Their observation was that if bacterial cell exposed to lactose, mean if lactose is present in the environment of bacteria, external environment of bacteria, then there will be increase the level of lactose utilizing enzyme. If more lactose is present in the bacterial environment, then there will be increase the level of lactose utilizing enzyme by 1000 to 10,000 times, 10,000 folds. And this increase the activity of this enzyme was due to increase the synthesis of enzyme, mean the enzyme is synthesizing more. So if the lactose is not present in their environment, then this cause abrupt termination in the synthesis of that enzyme. So that was the observation of Jacob and Monod for the lactose metabolism of bacteria. This whole phenomena is called enzyme adaptation, and on the basis of enzyme adaptation we will study the basic example of gene regulation in bacteria, and that will be the regulation of Lac Operon. Now we will see that what is Lac Operon and how it is controlled by some activator and repressor proteins. So what is Lac Operon? Lac Operon. In bacteria, few genes to be arranged together in the operon. What is operon? What is the definition of operon? Operon is a group of two or more genes under the transcriptional control of a single promoter. You know, the genes which are transcribed, the transcription is started by some sequence of gene which is called promoter and ends with the sequence of gene which is called terminator. Mean a promoter is a sequence of gene, a sequence of DNA nucleotide which start the transcription, and terminator is a sequence which terminate the transcription. So in one promoter there will be the transcription of two or more genes. In this diagram you can see that gene 1, gene 2 and gene 3, they are transcribed by the initiation of one promoter sequence. So this type of genes are called operon, which are under the transcription control of single promoter. And when transcription occurs, the messenger RNA which is formed, this messenger RNA contains the information of two or more genes. This type of messenger RNA are called polycistronic RNA. So this is the basic diagram of any operon. We will study here Lac Operon. What is Lac Operon? Now what is Lac Operon? This was the definition of operon. And what is Lac Operon? In bacteria, Lac Operon encodes a protein that involved in the lactose metabolism. Lac Operon will be specific for the lactose metabolism. So Lac Operon. Now focus to this diagram. This is the basic diagram of Lac Operon. In the previous diagram was just for an operon, and this diagram is an Lac Operon. You know what is Lac Operon? Lac Operon is the operon, the genes which controls the metabolism of lactose. Lac Operon, it consists of two transcriptional units. First is Lac Operon, which is starting from here, and second is regulatory gene, a basic simple regulatory gene, but Lac Operon starts from here. These are two different regulatory units. Lac Operon starts, you know, every gene is start from a promoter and ends to a terminator. Promoter is the sequence where RNA polymerase is attached and starts the transcription, and terminator is the gene sequence where RNA polymerase dissociate from the gene and terminate the transcription. But before Lac promoter, this is the sequence of promoter. You can see in red color. This is the gene sequence which is promoter and represented by Lac P, mean Lac promoter. Before this sequence, another gene sequence is present called cap site. What is mean? This is the gene sequence which is recognized by catabolic activator protein, a protein called catabolic activator protein. This protein binds to this cap site and help in the regulation of this whole process. Third gene sequence is operator site, Lac O, represented by Lac O, and this is called operator site. This is the gene sequence where repressor protein binds. Where a repressor protein, you have listened the name of repressor protein in the previous video in the regulation of gene. So a repressor protein binds to this operator site. After this, there are three genes, Lac Z, Lac Y, and Lac A. Lac Z gene encodes an enzyme beta galactosidase. Lac Y gene encodes an enzyme lactose permease, and Lac A gene encodes an enzyme galactosidase transacetylase. What are the function of these enzymes? The product of Lac Z gene, beta galactosidase, this enzyme that cleaves the lactose into galactose and glucose, and this enzyme also converts small percentage of lactose into allolactose. Allolactose is another regulatory molecule which helps in the regulation of transcription. We will see next. Lac Y gene, this portion of gene encodes an lactose permease enzyme, and this lactose permease is a membrane protein that required for the uptake of lactose from bacterial extracellular environment into cytoplasm. Mean when lactose is present in the bacterial extracellular environment, then bacteria uptake that lactose from environment and bring into its cytoplasm. So what is the protein? The protein that requires for the uptake of that lactose, this protein is called lactose permease protein. So for that protein, Lac Y gene is responsible. And the third and last gene of the operon is Lac A gene, which encodes an galactosidase transacetylase. This enzyme, an enzyme that covalently modifies the lactose and lactose derivatives, lactose analose. This enzyme just modify the lactose, and at last there is a terminator that is called Lac terminator, where RNA polymerase dissociate from the DNA and transcription stops, transcription terminates. So this is the transcriptional unit of Lac Operon. The second transcriptional unit is regulatory gene, and it is called Lac I gene. You can see here in green color. This is Lac I gene. It consists of its own promoter sequence, I promoter. There is a small promoter sequence and a Lac I gene, a small gene. This is not the part of Lac Operon. Lac Operon starts after Lac gene, but this gene have influence to the Lac Operon. Mean when this Lac gene, when transcription occurs here, then a small messenger RNA is formed. Let's suppose this is a messenger RNA. This messenger RNA encodes a protein called Lac repressor protein. This Lac repressor protein, when transcription occurs here only in regulatory gene, then a product repressor protein is formed. Then this repressor protein will bind to the operator site of the Lac Operon. This repressor protein will bind to the operator site of Lac Operon to control the transcription of Lac Operon. Mean when this repressor protein will bind to the operator site, it will stop the transcription of Lac Operon, and when this repressor protein will not bind to the operator site, then transcription will start of Lac Operon. So this is an separate part of which is present before the Lac Operon. These are two transcriptional regulation units are present in the Lac Operon gene. So this was the diagram of an Lac Operon. Now we are going to study that how this operon is regulated, which proteins involves in termination of Lac Operon gene, and which proteins involve in synthesis in increase the transcription of Lac Operon. So after the structure of Lac Operon, now we are going to study the control of the regulation of Lac Operon transcription, Lac Operon mechanism of transcription. Lac Operon is regulated by three ways: by repressor proteins, by activator proteins, and by operator sites. We will study them one by one. Our first mechanism is regulation of Lac Operon by repressor protein. What are repressor proteins? You are already familiar with the repressor protein, that the proteins that inhibit the rate of transcription, that depress the rate of transcription, that stops the rate of transcription. So when a repressor protein will form, and when it stops the transcription, and when it is not formed and it do not affect the transcription. There are two possibilities. When no lactose present in environment, if there is no lactose in the bacterial external environment, then bacteria have no need to transcribe the Lac Operon, because bacteria do not contain lactose in the environment. So there is no need of the enzyme that metabolize the lactose. So what is the procedure to stop the transcription? We have seen that there are two regulatory sections. First is Lac Operon, the second is Lac regulatory gene. This Lac regulatory gene, first of all, transcription occurs here. When transcription occurs here, a messenger RNA is formed. This messenger RNA encodes a protein called Lac repressor protein. This is a Lac repressor protein, a homotetra molecule. 4 molecules of proteins are arranged to form a large molecule. When Lac repressor protein is formed, this Lac repressor protein will bind to operator site. This is the operator of Lac Operon. So when this repressor protein will bind to operator site, this is an operator site, and this repressor protein will bind to operator site and it will stop the transcription. So no messenger RNA synthesis occurs, mean no transcription. In this way, bacteria stops the transcription of Lac Operon, because bacteria have no lactose in their environment, and bacteria do not require the enzyme for the metabolism of lactose. Now the second category is that, the second phenomena is that when lactose is present in the environment, when lactose is present, this, for example, this is bacteria and lactose is present in its environment, then bacteria uptake that lactose in its cytoplasm. This lactose will change into allolactose. the function of beta galactosidase enzyme. Now the lactose is present in the environment and bacteria need the enzymes to metabolize the lactose. So now transcription occurs and the gene will produce its product. So how transcription occurs? Now lactose is present, it will convert into allolactose, and the allolactose molecule which are present inside the bacterial cell, these allolactose molecule binds to this repressor protein and a conformational change occurs in this repressor protein. These allolactose molecule, they bring a conformational change in this repressor protein, and this repressor protein will not bind to operator site. Now this repressor protein will not bind to operator site, and when this protein will not bind to operator site, then RNA polymerase attached here and transcription starts and messenger RNA synthesized. This messenger RNA now produce their products, and these enzymes are now produced to metabolize the lactose. So this is the mechanism of regulation of Lac Operon by repressor protein. Repressor protein, when no lactose is present, what is the summary? When no lactose is present in the environment, then this repressor protein will bind to operator site and inhibit the transcription. But when lactose is present in the environment, then that lactose brings into the cell and converted into allolactose. This allolactose molecules bind to the repressor protein and brings a conformational change in the repressor protein, and this repressor protein will not bind to operator site and transcription starts. And when transcription starts, then the enzymes to metabolize the lactose, their synthesis start. Now the second mechanism of Lac Operon regulation is the regulation of Lac Operon by activator protein. Now the situation is different. If there are two substrates present in the environment, glucose and galactose, then if two substrates, glucose and galactose, are present in the environment, then bacteria will first uptake glucose into the cell and metabolize the glucose. Due to glucose, bacteria will not uptake the lactose and will not transcribe the Lac Operon. But when glucose is used up, then Lac Operon is transcribed and lactose metabolism is start. First, bacteria will metabolize glucose. This is called diaxic growth. Now there are four situations for bacteria. If in the environment lactose is present and no glucose is present, then what will be the transcription? We will see in detail. And second category is that no lactose is present but glucose is present in the environment and no lactose is present, then what will be the transcription? The third category is if lactose and glucose both are present, then what will be the transcription? And D is that the fourth category is if glucose is present in the environment and no lactose is present in the environment, then what will happen? What will be the transcription? We will study these four mechanisms, these 4 points one by one. Now what is the basic mechanism of gene regulation by activator protein? There is an activator protein called Catabolic activator protein, CAP, which is bind to cap site. Catabolic activator protein site. Catabolic activator protein site is the gene sequence where catabolic activator protein binds. And another protein called CAMP, adenosine monophosphate, cyclic adenosine monophosphate. This is produced from ATP, and this is a regulatory molecule that regulates the catabolic activator protein. So this is the protein that control the Lac Operon. What is the mechanism? We will see these four mechanisms one by one. So there are four mechanisms, there are 4 points when glucose and galactose both are present in the environment. The first, when only lactose is present in the environment and no glucose is present in the environment. There is no glucose, just lactose is present in the environment. When lactose will present in the environment, then this protein C cyclic adenosine monophosphate, this protein will be in high concentration. When this protein will be in high concentration, this will bind to CAP protein, catabolic activator protein. Then this catabolic activator protein will bind to CAP site, and when this catabolic activator will bind to CAP site, there will be high rate of transcription. Messenger RNA will be synthesized and there will be high rate of transcription. I am repeating that when lactose is present and no glucose is present in the environment, then there will be high catabolic cyclic adenosine monophosphate. Due to high concentration of this protein, this will bind to the CAP protein, then CAP will bind to the CAP site and the rate of transcription will be high. And in second category, when there is no lactose is present and no lactose and galactose, when there is no lactose and no glucose, when there is no lactose and no glucose is present, then the repressor protein, which we have studied in the previous point, this repressor protein will bind to operator site, and it will block the transcription, it will reduce the transcription, and there will be no transcription. B point, what was the point? When there is no lactose and no glucose is present in the environment, then there will be no effect of this protein. Then repressor will bind to the operator site and it will stop the transcription and there will be no transcription. So there is the third scenario, the third situation C is that when there is both lactose and glucose both are present in the environment, both glucose and lactose are present in the environment, then what will happen? When both lactose and glucose are present in the environment, then lactose inactivate the repressor, lactose will inactive the repressor by the binding of allolactose, mean now repressor will not bind to the operator site, and due to presence of glucose there will be low cyclic AMP. What will happen when both lactose and glucose are present? Lactose will inactivate the repressor by binding of allolactose, and due to glucose there will be low cyclic AMP. When there will be low cyclic AMP, then this cyclic AMP protein will not bind to CAP protein, CAP, catabolic activator protein. When this cyclic AMP will not bind to CAP, then this CAP protein will not bind to CAP site. It will not bind to CAP site. So this was protein that was increasing the transcription. Now this protein is not binding to CAP site. Then transcription will occur but in slow rate. Transcription will occur but in very slow rate. Messenger RNA will form but the rate of transcription will be very slow. This was the third category, third scenario. The fourth situation is that if glucose is present in the environment and no lactose is present, then what will happen? The presence of glucose, due to the glucose there will be low cyclic AMP, there will be low cyclic AMP. This protein will be low and this protein will not bind to the CAP protein. Due to glucose there will be low cyclic AMP and less cyclic AMP. Due to less cyclic AMP, this protein will not bind to CAP protein, and no lactose, glucose is present and no lactose, there is no lactose. Due to absence of lactose, this repressor protein will bind to operator site and it will stop the transcription. So when glucose is present in the environment and no lactose is present, then there will be no transcription. So there are these 4 situations of the regulation of Lac Operon by activator protein. What are these 4 situations? When lactose is present in the environment and glucose is not present, then transcription rate will be high. And when lactose is not present, lactose or glucose both are not present, then transcription rate will be very low or there will be no transcription. And when lactose and glucose both are present in the environment, then transcription rate will be low. And when only glucose is present and no lactose is present, then transcription rate will be very low or there will be no transcription. This is the mechanism of gene regulation by Lac Operon regulation by activator protein. We have seen the repressor, Lac Operon regulated by repressor protein, and now we have seen the Lac Operon regulated by activator protein. Our third mechanism is the regulation of Lac Operon by operator site. Now our last and third mechanism of regulation of Lac Operon is by operator sites. What is operator site? Remember that when we were studying the structure of Lac Operon, we said that there is a gene sequence called operator site to which a repressor protein is bind. You know what is repressor protein? Repressor is a protein that inhibits the transcription, that stops the transcription. So this repressor protein is bind to operator site and it stops, it represses the transcription. Further genetic and crystallographic studies have revealed that there are 3 operator sites present in the Lac Operon. Operator site 1, operator site 2 and operator site 3. Operator site 1 is the main operator site where repressor protein will bind. Operator site 2 is present after the LAC-Z gene and operator site 3 is present before the operator site 1. These 3 operator sites have role to inhibit the transcription, to repress the transcription. You know that repressor protein will bind to the operator site. In the experiment, there were 1300 strains were studied. If there are 8 categories, if 3 operator sites are active, mean all the operator sites are active, then repression will be in all 1300 strains. Mean if there are 1300 strains of bacteria of genes, if all three operator sites are active, then the repression will occur in all strains. But if operator site 2 is missing, mean there is mutation and operator site 2 is not working, only 1 and 3 are working, then the repression will reduce to 440 strains. And when operator site 3 is missing, only 1 and 2 are present, then repression will be in 700 strains, mean it is reduced. And when operator site 1 is active and 2 and 3 are missing or mutant or missing or not present, only operator site 1 is present, then the repression will be dramatically decreased to the 18 strains. And when the operator site 1 is missing, both 2 and 3 is present, the repressor will only be in 2 strains. It is dramatically reduced to 1 or in 2 strains. In both of these you can see that operator site 1 is missing and 2 is missing, repression will only in 1 strain. If 1 and 3 both are missing, repressor will be in only 1 strain. And if all three operator sites are missing, then there will be no repression only in 1 strain. Repression is only in 1 strain. It means that operator site 1 has the major role to repress the transcriptional process. These are the 3 mechanisms to control the Lac Operon. The first mechanism was the Lac Operon regulation by the repressor protein. The second mechanism was the regulation of Lac Operon by activator protein, and the third mechanism was that regulation of Lac Operon by operator sites. So this was the whole mechanism of regulation of Lac Operon in bacteria.